I am really happy to be here. As you've seen, for the families who may be watching, this is really complex. And as scientists, as this amazing group that I'm so privileged to be a part of, we are trying to work on two areas. So the first is to find a drug, as we were just discussing, the rapamycin or maybe something else, a drug to help the kids with their symptoms. The second thing is to go in and do gene therapy, gene editing, gene modification, whatever we can do when we really understand what we need to target. And so while we're figuring all this out at the molecular level, while the people more brilliant than me are figuring that out, the gene therapy and cell therapy and gene modification fields are rushing forward. So I'm going to give you a little update on that, and we're hoping to get here. So healthcare is really changing. Stem cells, immunotherapy for cancer patients, gene therapy, gene editing, regenerative medicine. And these new frontiers involve living medicines. So cells, modified viruses, other products that are not pills, it's not a lyophilized vial in your pharmacy. They're things that we need healthcare teams to produce, formulate, and deliver to the patients. And this is a whole new field of healthcare, and it's the one in which I've worked now for 30 years and is finally coming to fruition, where we're seeing some actual medicines that can be prescribed, living medicines that can be prescribed for kids and adults. So I'm very lucky to direct the UC Davis Stem Cell Program. We've had funding for facilities, team training, and research from the California Institute for Regenerative Medicine, or CIRM, NIH private donors, foundations, and California State is actually giving the allocation for some of this research that's going on here, large portion of this research. Very fortunate to be a part of that. We have about 24 stem cell and regenerative medicine trials ongoing at UC Davis and about 20 in the pipeline. Those with the green arrows are the clinical trials that are ongoing. Just to show you how broad our teams of experts are. And the red ones, we are talking to the Food and Drug Administration about starting a new clinical trial, and talking to them means submitting a whole bunch of paperwork showing that it will be safe and hopefully effective. That's where we'd like to get with this, yes.
What kind of timeline did these projects?
They're all different. Some are quicker if they kind of piggyback onto another trial, if they use the same type of cell. It can be just a year or two. Others have taken, 10 years. So it just depends. Our goal is to move this into one of the pipelines, test on the mice, get the data, and move into one of the pipelines once we understand exactly what we should target. And it's hard to know the timeline, but yeah. And hopefully, meanwhile, we've developed it, understood, and the kids have some drug that can help with the symptoms while we're working on these more permanent treatments. So these living medicines are made in good manufacturing practice facilities. We're very lucky to have one at UC Davis. This is a cellular and gene therapy product manufacturing for clinical trials that have US FDA approvals. We don't do anything without approval from the FDA. We have 6 individual suites, so 6 patients can be, their cells or gene for them can be being made at the same time every day. We have 2 shifts going now. And we're working with a lot of academic and industry partners. We use a hollow fiber bioreactor to make vectors, and I'll show you what the vector is in just a minute. And this is for gene therapy, so we can make pretty large scale. This is a machine. The cells making the virus, modified virus for gene therapy, are in there. This is their food, this is their waste, and it actually calls you when it wants one of the bags changed. It will ping you on your cell phone. So it's this field is getting really automated. And we have a clinical team now to deliver the clinical trials, our Alpha Stem Cell Clinic. This is a group of wonderful people funded by the California Institute for Regenerative Medicine. And there are 5 of these sites throughout California. And you can call us to get referred to a trusted gene therapy or cell therapy clinical trial for the indication. So gene therapy is coming of age. It's been a long time coming. Started in, that's 1960, down at that end there. I came into the field about here, worked on a lot of this, getting the vectors, and gene transfer to mice and working out how to get the genes into the human cells to do the gene correction. And meanwhile, the amazing MDs with whom we were working on how to do conditioning for the kids, to get the gene therapy to them, and finally how to work on CNS disorders here, and now to do gene editing. And so we'll be hearing a lot more about that as we go. So there are two main types for gene therapy, the lentivirus or lentiviral vectors, and these are, and then the adeno-associated virus or AAV. And you'll hear about both of these for the families. If you're on Facebook, you might see other families getting these therapies. That's because groups have been working on them, like we're working toward the therapies for this one. So these viral vectors, they carry the gene of interest to the patient. Basically, we've taken a virus that will go into the patient's cells. We've completely gutted it, and we've put in the gene that we want to carry. So the cells, for lentiviral vectors, the cells will be removed from the patient and modified ex vivo out of the body in a GMP facility, like I showed you that we have, maybe in one of those machines that calls you, and then returned to the patient. AAV is very interesting. They can be delivered straight into the bloodstream or straight into the target tissue like the brain. And these are really cool vectors and in development now for many indications, as I'll show you. And this just shows again the direct delivery using the AAV. We can package the therapeutic transgene once we know exactly what would be best for this indication, package it into a delivery vehicle such as a virus, and inject into the patient for liver, brain, other organs to target. So the first AAV-delivered gene therapy for an inherited disease approved by the FDA was in 2017, and this was for a rare inherited blindness disorder, and this is directly injected into the eye, so that was like a safe, contained space, shown to be efficacious. And when it gets approved by the FDA, that means that the MDs can prescribe it and it can be covered by insurance, and this is very important for therapies to get to that stage. The second one, Zolgensma, is a landmark AAV-delivered gene therapy to treat spinal muscular atrophy, which is a terrible disease that affects kids, and this has been approved just earlier this year. This latter one depends on, hard to read this, but depends on this AAV9. This is a type of the AAV vector that can be injected into the bloodstream and can cross the blood-brain barrier to go into the brain. And that sounds wonderful for our indication, but we have to be very careful with it because if you're using very high dose, there has been some toxicity in animal studies. So people are working out these details while we are working out the specific gene to put into these vectors, and that's the paper describing that in primates and piglets. Meanwhile, patients in clinical trials have been dosed with this AAV9 without seeing similar problems because they're being dosed at a lower dose. So we have to understand how to use these vectors so that they're safe and efficacious for the patients, and be very careful. And the first patient has been treated with gene editing. This was back in November. It was for targeting to the liver using AAV by the company Mesenchymal. And so this has begun, and we would certainly like to get there in the future. So different options with therapeutic stem cell and gene therapy. Gene therapy, what I was just describing, this adds a normal gene copy to restore function. And the hematopoietic or blood-forming stem cells are currently best for long-term therapy because you do basically a bone marrow transplantation, and then all the daughter cells from those engineered cells will have the new gene. Cell replacement strategies we can think about for the future. This is transplantation of healthy progenitors capable of differentiation into the tissue of interest, and transplantation of gene-modified patient induce pluripotent stem cell-derived cells. This really, to me, is the farthest off. We would like to do the gene therapy first, and that's because in the brain, the neurons we're just at the beginning of understanding how we can transplant cells into the brain and have them functionally integrate into the brain. But there is a lot of work going on in this area. So tissue healing, promoting angiogenesis and neurogenesis. A type of cell that I'll show in just a moment, mesenchymal stem cells. They secrete factors that promote healing over inflammation and scarring and are easily gene modified. And we use them as a kind of cargo ships. They deliver things into the body. And then finally, gene modification and correction. This is really what we want to do. We want to silence the mutant gene so that it's not directing the cell to do things that we don't want it to do, or edit the gene for a dominant mutant allele. That's one that is out of control, and we want to shut it down. And these mesenchymal stem cells can carry cargo, and Dr. Fink's group is working on that. So this is the cell-based therapy. We take cells out of the body. The therapeutic transgene is packaged into a delivery vehicle, such as a viral vector. Therapeutic transgene goes into a delivery cell, such as a stem cell, that is often derived from the patient. And then these cells are multiplied in the GMP facility and then readministered to the patient. So I'll just go through two types of the cell-based gene therapy. So there's the bone marrow stem cell gene therapy. This is the first one that's been around. And for this one, the stem cells are taken. Here's the virus. We disable it. We take out all of its genes, and then we put our genes in. Almost all of its genes are out. It can't cause harm. We package the gene that we want to transfer into this viral vector, put that in the cells. This is called transduction. And then the corrected stem cells are transplanted into the patient. I started my career working on bubble babies 30 years ago with Don Cohn, who was an amazing mentor. And Don, I went off to have my career and work on other cell types. Don kept working on this disorder and has now functionally cured over 30 kids with this disease. And bubble baby disease is where the kids are born with a mutation in the enzyme adenosine deaminase, and that means that they functionally have no immune system, and so they would have to live in a bubble. I don't know if any of you are old enough, like I am, to remember the movie The Boy in the Plastic Bubble with John Travolta, but that was that disease. And so he had to live in isolation because he had no immune system. And that is this disease. But now, we as scientists seldom use the word cure, but this actually has been a cure for these kids because they were deficient in one enzyme that got put into their own bone marrow, transplanted back into them. They got some ablation and knocked out a lot of their bone marrow so that they could get the new corrected bone marrow. And this is little Evie, and she is cured. She used to have no immune system, and now she's going to school and in public pools and functionally cured. And at least 30 of the kids are now functionally cured. So this is one type of gene therapy that actually is working and is going forward for many different disorders. Now, how to get things into the brain, that's different. It's harder. Of course, it's harder. But we do know that microglial cells in the brain come from hematopoietic or blood-forming stem cells, just like I had described for the last disease. And so the bone marrow transplant with gene therapy has a chance to impact some diseases that affect the brain. And so these cells are coming from the bone marrow and going into the brain. And we have teams working very actively on this. And there's been a clinical trial, hematopoietic stem cell gene therapy for cerebral adrenoleukodystrophy. And the conclusions were that it was safe and the boys had improvement, and additional later phase clinical trials are indicated. So we have teams working on hematopoietic stem cell gene therapies for many different diseases, and moving forward for those. And by the way, our other teams, we all work together in our program, and if they make an advance and learn something, then it really helps us. It helps this team, and our kids. So 87 clinical trials for hematopoietic stem cells and gene therapy. That's moving right along, and here are our groups who are working on that type of gene therapy. There's another type of cell that I had briefly mentioned, the mesenchymal stem cell. These are paramedics from the bone marrow and also from fat. We can get them from our spare tire. These are the broad, flat cells that support the hematopoietic stem cells in the bone marrow. They are great for tissue repair, and they are really great delivery vehicles. So we use them to get things into other cells. And so they're a candidate for delivery of gene editing tools, and this is all I'm saying about this because it's really Kyle's work, and I don't think he's updating on it today, but his team is working very hard on this. So these cells secrete factors, reduce cell death, enhance connections, and they can be readily engineered using viral vectors to robustly deliver cargo. They remain in the brain for 1 to 2 months, the way that we condition them, and they are migrating around during that time. They don't require immunosuppression, strong safety profile, and they've demonstrated delivery of intracellular contents into neurons and other cells. And this is a little video showing the mesenchymal stem cells in red, and these are some damaged muscle cells in green. And I don't know if you can make it out, but the mesenchymal stem cells are making they're delivering these little packages to the other cells. There's a green cell right here. Red package goes into it. They're delivering the contents of themselves, basically, into those other cells. So that's RNA and protein. And when we mix them into a tissue, they're delivering stuff from themselves all over for about a month, and then they go away. And they can also deliver mitochondria, which is very interesting. These are the little circles in this video going into the target cell. And so it's a temporary kind of delivery truck to get our cargo into the tissue of interest, and hoping to further use these to deliver things into the brain. A lot of clinical trials using mesenchymal stem cells. They're very safe. They don't hang around forever, and our teams are working with them a lot. So almost last slide. Challenges for treating these single gene disorders in the brain through gene editing. It's delivery. It's all about location, location, location. So how can we get these gene editing molecules into enough neurons or neural progenitors in the brain to really have an impact in the person? Do we use the AAV that I talked about at first, other vectors like the lentiviral vectors, artificial delivery methods, nanoparticles, liposomes, MSCs, exosomes that come from them, those little particles. So that is a big focus of Dr. Fink's lab, and he said he didn't have time to cover it today, so just wanted to mention it. And finally, in this new era of living medicine, cell and gene therapies are already being prescribed to treat diseases and injuries. And so this is where the field's going. We hope that this is where we will go in the future once all of the science is worked out. These are things that are approved now, and that means that the clinicians can prescribe them, and they can be reimbursed by insurance. So everything takes teamwork. I'm just very, very grateful to be a part of this amazing team, and also to interact with all of the families on Facebook. And it's all for the kids. It's all for the patients. So also on Twitter. I tweet a lot.